Today's talk is titled Rebuilding the Tumor Microenvironment for In Vitro Discovery. I'm Meredith Osborne. I'll be starting the presentation and then handing the floor over to Dr. Sean Fall, who will continue. But to set the floor for why rebuilding the tumor microenvironment matters, first we're going to look at clinical trial success. So starting on the right side of this diagram, and here we're focused specifically on oncology clinical trials, only 5.1% of oncology drugs coming out of clinical trials succeed. And in order to succeed, right, we were talking about that the data is good in patients. The drug is effective and it is safe. Of course, the flip side of that is that nearly 95% of drugs going into clinical trials fail, either on the efficacy or the safety front, or both. And as you're likely aware, clinical trials are an incredible investment of money, time, resources, and patients themselves volunteering to participate. The cost of developing a new drug is $172.7 million, but then increases to $515.8 million once you add in the cost of all of these failures. So in order to justify going into a clinical trial in the first place, you have to build a really strong case for the hundreds of millions that are going to be spent, for the years of time that are going to be invested, and for the patients themselves who are going to participate in receiving this experimental therapy. So it is safe to say that 100% of drugs going into clinical trials have really strong lab data. And yet there's this huge disconnect, right, between really, really good lab data to justify the trial in the first place, and then this really incredible failure rate and very, very low success rate coming out of clinical trials once we have done that testing in people. And in the industry, this disconnect is called the translational gap. And it is nothing new. We have all been well aware of the translational gap for quite some time and have been building technologies intended to address and speak to this gap. And as those technologies have improved, at the same time, the complexity of the therapies we're offering has also increased. And you pair those two forces with the fact that this is frankly just a difficult problem to solve. And you have this exceptionally persistent translational gap that we have not been able to address. And when we talk about addressing the translational gap, we're really looking here at the beginning. What are we doing in the preclinical lab research to generate that good lab data that we hope will be better reflective of how this drug will perform in actual patients? And the backbone of preclinical lab research is tumor models in the oncology space. So I'm going to talk us through several kind of key categories of tumor models, and then hand the floor over to Dr. Sean Fall, who will address co-culture strategies for recreating the tumor microenvironment, specifically with a tumoroid tumor model approach. So the first tumoroid we'll be discussing is cell lines. Cell lines are one of the oldest tumor models. The first human cancer cell line was discovered in 1951 using cells from the patient Henrietta Lacks. Obligatory shout out if you have not read the book, The Immortal Life of Henrietta Lacks. I highly recommend it. It's a personal favorite. But cell lines are clonal, meaning the cells are all genetically identical to each other, which is not how real tumors are in humans. They're very genetically different. They're 2D, which again is not how real tumors are in humans, but these are human tumor cells. And the real strength of cell lines is that they are extensively characterized in the literature. Their genomic expression, their phenotypic presentation, their drug response data. There's just a vast wealth of characterization data available for nearly all cancer cell lines. Suitable uses for cell lines. They are broadly available and they're easy and inexpensive to culture. So they are great for initial pilot studies, but specifically for drugs that are intended to have a direct impact to tumor cells. Cell lines are tumor cells and they are only tumor cells. So any drug that relies on a cell-cell interaction, as many of the more complex therapies that we're seeing in research today are, cell lines are not suitable even for this kind of initial quick and dirty pilot. The other great suitable use case for cell lines is as controls. Here at Discovery Life Sciences, we do a number of receptor cleavage studies to test whether a biomarker, how it's impacted in our dissociation protocol, and we've published that data in our DCC user guide. And again, this is relying on this extensive characterization. If some new biomarker is, kind of newly of research interest, we don't know how our dissociation protocol impacts that biomarker. We can go out and find cell lines that are known to express it, bring those in, run them through the protocol, see what happens, right? And that's just an example of how cell lines will remain part of our research as really excellent controls. The challenge with cell lines, though, is they have very, very low clinical predictivity. As I said, these have been around since the 50s, have been incredibly useful for scientific aims, but have not been able to, close that translational gap because they do not predict well how a drug will behave in a whole organism, a full person. And like I said earlier, they are unsuitable for drugs that rely on interactions between a cancer cell and a different cell in the body. Which brings us to our second tumor model, which is animal models. Animal models are genetically engineered or xenografted animals. So these are not human, these are animals. Research programs tend to start with mice. And then as they are, building that case, generating data to get closer to ultimately justifying moving into a human clinical trial, and the pharma industry moving into beagles or monkeys are kind of two of the more common, fairly advanced animals that you see. But mice are the foundation, the mainstay of animal model research. Animal models include a heterogeneous cell population, right? Unlike the clonal nature of the cell lines. Again, unlike cell lines, they're 3D, kind of by definition, right? A whole animal. And the real strength of animal models is that they represent the whole body system. So you have the tumor in the environment with a functional nervous system and a functional circulatory system, functional, total immune system. And so that's the real strength. Previously, animal models have really kind of been our best solution for drug response. Tumor size, for example, is a key data output, right? We give the animal the tumor, then we give it our drug that's intended to treat the tumor, and we see how big the tumors are. Do they grow larger? Do they stay the same? Do they shrink? However, currently, we are beginning to see really kind of at all levels, including a bill that was just passed in the Senate earlier this week, FDA Modernization Act 3.0, a move to phase out animal models in favor of what the US FDA is calling NAMs, New Approach Methodologies. But similar moves are being taken by regulatory bodies in the UK and Europe and other countries throughout the world. And this is really recognizing that while animals have been, kind of our best, most advanced technology, and were needed in order to justify moving into human research, that we now have other options, other new approaches, 3D cultures such as tumoroids, which will be the heart of today's presentation, as well as in silico models, which are purely digital AI machine learning type models that are now advanced enough and able to replace much of what animal models have done in our drug discovery work. And that shift, moving away from animal models in favor of new approach methodologies, really addresses the main challenges. Animal models, the biggest problem is the species difference. There was this incredible quote by Dr. Richard Klausner, who was the National Cancer Institute director in the late 90s, that cancer research has been a long history of curing cancer in the mouse. We have cured cancer in mice for decades, and it simply hasn't worked in humans, right, because of those species differences. And then, of course, there's also the ethical concerns of working with animal models that the new approach methodologies are able to, move away from. Next tumor model that we'll be discussing is fresh tissue. These are surgical patient tumor specimens. So unlike animals, they are human. Unlike cell lines, they are heterogeneous and they are 3D, and they have all cells present. One of the weaknesses of fresh tissue, though, is there is no functional vasculature and no functional nervous system. Any blood vessels or nerves, that had infiltrated the tumor are present, but they're not hooked up to anything anymore, right? Because this is a surgical specimen that was surgically removed from the body. So that function is lost. Suitable uses for fresh tissue, they are used for drug testing assays, notably short-term drug testing assays, because you only have a certain window of viability for fresh tissue and can only do assays that can be completed within that time frame. And an example, organ-on-a-chip systems, which to grossly overestimate a engineering marvel, they are essentially very, very fancy Petri dishes that you put your fresh tissue into along with some other inputs, and it's intended to help try to replicate those missing whole body functions of, say, vasculature, for example. The challenge with fresh tissue, it is fundamentally impossible to standardize, right? So unlike a cell line where, if I read a paper and it's super interesting and they use cell line 1, 2, 3, 4, I can go out, acquire cell line 1, 2, 3, 4, replicate the study and expect a similar response. Fresh tissue is not standardized like that, right? If you read a really great study that used fresh tissue, you cannot get that exact same fresh tissue from that exact same patient. No one can get that again, right? That was kind of a one and done, and every tumor is different and every patient is different. So we represent the real world's diversity at the expense of it is impossible to standardize. Tumor tissue also faces challenges of scale. This is the business that we're in at Discovery Life Sciences, and there are fundamental limits to how much you can scale up a fresh tissue operation, so that's a challenge. Viability, as I spoke to, right, once the blood supply is cut off during the surgical procedure, your ticking clock has started as far as how long this tissue will remain viable. And so you have to get through all of your operational hurdles, right? Get it from the operating room out to, and into the researcher's lab and give them time to do their experiment within that kind of window of viability. And lastly, the challenge, there's a limited data for targeting collection. Most of the really rich, really useful data that we get from patient surgical samples happens after surgery. Writing the path report happens after surgery. Genomic testing may happen after surgery. Companion diagnostics may happen after surgery. And when they happen after, they are not available at the time of collection to make a decision about whether this meets the inclusion-exclusion criteria for a particular project. So that's kind of the final challenge with fresh tissue. And that brings us to our final tumor model for today, which is tumoroids. Tumoroids are heterogeneous 3D populations of human tumor cells. So right, unlike animal models, these are primary human patient tissue. It maintains that heterogeneous population, right? So unlike cell lines, which are clonal, all the cells are identical to each other, whatever genetic diversity is present within the tumor cells of the original tumor the tumoroid is derived from is maintained. And Dr. Sean, in his slides coming up, will be able to show how we measure that to maintain and confirm that that heterogeneity is still representative of the original tissue. The 3D structure, tumoroids are self-aggregating, so they form complex 3D shapes, again, more like how tumors actually present in the body. But they are tumor cells only, which is the main challenge then, skipping down to the bottom, is the lack of other cell types. So tumoroids are great for drug dose response curves, which is a kind of data readout that we're used to seeing from clinical trials. And so it's great to have a lab tumor model system that's able to give us that same data output. And the reason why they're so well equipped for drug dose response curves is that they are designed for long-term cell cultures, right? So unlike fresh tissue, which always has to be short-term, these can be long-term. Unlike animals, these are human, and unlike cell lines, these are heterogeneous in 3D. But as I said, the primary challenge working with tumoroids is the lack of other cell types, which if you remember back to the title of this presentation, it's all about recreating the tumor microenvironment in vitro. And so this is where I'm going to hand the floor over to Dr. Sean Fall, who will share more details on co-culture strategies to address those missing cell types.
Fantastic. Thank you, Meredith. So as Meredith was saying, one of the big focuses that we've had for this webinar and a long time at Discovery is the idea of the overall tumor microenvironment. And so this is a nice cartoon that we have, and Meredith had been showing this on some of the slides that she presented on. And so I just want to take some time here to discuss the microenvironment as a whole. You know, a lot of folks throughout the years we say the tumor, they think primarily of cancer cells, but for decades it's been realized that there's a whole lot more to a tumor than just the cancer cells themselves. There are the elements of what we call the tumor cell elements, like cancer-associated fibroblasts. Like Meredith mentioned, we have the blood vessels that provide nutrients and oxygen to the tumor. The neurons or the nerves as well that go through the tumor. And honestly, this is a cell type that I personally didn't think about much until I went to SITC this year, and there were sessions dedicated just to the idea of how to target neurons and nerves within the tumor for therapeutic benefit. So it just even recently, a lot of these cell types are being evaluated for potential targets. The other large component of the tumor microenvironment are those immune cells. And as you saw from my intro, I am an immunologist at heart, and I could spend the next week going over each of these and how they both will help the tumor evade the immune system, as well as how they will help kill the tumor, either with or without any sort of therapeutic intervention. So what we're going to do through the next couple slides is basically look at different aspects of the tumor microenvironment, how we can grow these in vitro, how we can hopefully start putting these back together to recreate this tumor microenvironment using the most physiologically relevant patient material. So we'll first start with the cancer cells. So this is where Meredith left us off, which was at tumoroid models. So for our tumoroid models, what we've been using is the OncoPro system provided by Thermo Fisher. And so these tumoroids, which you can see growing on the right side of the screen here, this happens to be one of the lung lines, are physiologically relevant. So this is initiated from primary patient material. They are three-dimensional, and they will self-aggregate. And what I'll show in some genomic data on the next couple slides is they are heterogeneous. We're not looking at any sort of single clonal population. What's very key for these tumoroid models, both as we initiate them and then as we expand them and use them, is that they are highly characterized. So we do some very deep profiling at the genomic level, both RNA and DNA. We have all of those pathology reports that Meredith had mentioned as well. Why this is very important is we are in the era of personalized medicine. What one patient receives versus another patient with the same disease is going to be vastly different based on their mutational profile or their biomarker or target expression. And so as we build out these models, since one of the methods we do this is using a cryopreserved starting material, we can get very highly specific as to what mutations we're looking for or what expression patterns we were interested in. Additionally, these are highly scalable, and so I'll go through how we build these, but you can get billions of cells from these models, which really makes drug testing very economical, as well as very scalable and easy. You don't have to worry about unique patient differences. You're essentially using the same patient material. They are stable, so again, we'll show some data on how even through late passages, these maintain that inherent genomic stability, and they're reproducible. They can be cryopreserved, reinitiated, and grown out, which is pretty much how we've been handling these by having certain banks of cells as we use different studies. So if we look at the various diseases or indications that we are currently using in the lab, we have those that have already been developed. We know the composition of the media to grow these. We have these cryopreserved and ready to expand or to use in drug studies, and those are largely tumoroids generated from breast cancer, colorectal cancer, endometrial cancer, and lung cancer. We have a second group, and these are our in-process group. So for the 6 indications that we're primarily looking at on the right side of the screen there, these are indications where we have a very good idea of what the various growth factors, cytokines, additional additives we need to add to the basal media to make these grow, but we're still doing some R&D to get the exact components, their levels that are needed. Or a corollary to that is just getting the tissue in to work with. Something like pancreatic cancer, which for us is rare to acquire. By the time we get it, a lot of times, because it's from the pancreas, it's dissociated itself. And so basically, how are we going to get this tissue and how are we going to use it as we develop these models? For the rest of the talk, I'm primarily going to focus on 2 of our models. So we're doing a side-by-side comparison of 2 donors. So I will refer to these donors by their patient ID. These are 9-digit codes that we use internally to keep our patients separate and know which samples come from which patient. For the rest of the talk, I'll use just the last 4 digits, 3309 or 1104, as a handy way just to be able to distinguish the 2. What we like about this side-by-side comparison is that these 2 patients actually have the same disease, colorectal cancer, same subtype, adenocarcinoma, slightly different stages, but from the same location, the colon. So clinically, very, very similar patients. One is male, one is female, and we do see some slight differences in terms of driver mutations. So our 3309 patient is a G12C KRAS mutant donor versus a wild type KRAS for 1104, but largely donors that are very similar. And what we'll see as we move through this is that even though these are similar clinically, they are significantly different genomically, proteomically, as well as the response to compounds. So the way that the tumoroid development works is that we do start with a single cell or a near single cell suspension from a primary patient sample. Because we do want to focus heavily on getting highly characterized samples into the system, the majority of how we initiate these does come from a cryopreserved bank. So you'll see me use the word viably cryopreserved cells as we go through the rest of the talk. However, for some indications, particularly something like pancreatic cancer, we are also using fresh tissue out of our European network to initiate these tumoroids as well. These single cell suspensions are initially propagated through an embedded stage. These are kind of the more traditional tumoroid or 3-dimensional culture systems where the cells are embedded within a 3-dimensional matrix. It forms a little bit of a dome on the bottom of a tissue culture vessel. These are great. They really allow cells to aggregate, to start to grow clonally. But they do have a limit in terms of scalability. Dealing with lots and lots and lots of domes can be very, very difficult and require a lot of hands-on experience from very highly technical technicians. So the nice thing about these tumoroid systems is that after the initial growing in this embedded stage, they're transferred to a suspension culture stage. And this is really crucial because this allows for the scalability of these systems. I like to think of the suspension stage, even though these are solid tumor indications, they look a lot at this stage like kind of a leukemia or a lymphoma cell. They grow in clusters, they grow 3-dimensionally, so you won't see them within just the dome. They are throughout the entire 3 dimensions or the Z stack of the culture, so you get a lot of cells generated. And so what we do is during these 2 stages, we will take and freeze back a certain proportion of these cells before we initiate the next stage. And so this gives us a ultra low passage frozen bank. And this is crucial because as we generate a cryopreserved bank that we can use for studies, this is a mid-passage bank that will be depleted. And so what we can now do is go back to our low passage suspension bank, expand it out, do a lot of QC metrics to make sure that it still represents the patient tumor, freeze it back. If this is depleted, we go back to the embedded stage. And so it gives us a lot of redundancy and a lot of ability to basically make these tumoroids last as long as we can because of their value as primary patient material. So one of the biggest questions, obviously, when dealing with a system like this is, are you still representative of that parent tissue? If you're not, then the idea of this ex vivo system kind of falls apart. It doesn't really represent patient biology. So this is donor 3309, and what we're looking here as whole exome data. So we're looking at the variant allele frequency, or the VAF, across all of the SNPs, all the variants that were found in this study. On the top panels here, we're looking at that initial viably cryopreserved tissue, and then comparing it to the genomics of the embedded, the suspension, or the working bank. The working bank is that cryopreserved bank that we've generated. And so comparing back to our viable cryopreserved tissue, so this is the initial patient tissue, we see a high degree of correlation between the VAFs from each of these banks to that parent tissue, R-squared values of 0.86, 0.87 and above. As we move to the embedded stage, now if we look at this embedded stage compared to the suspension and working, these R-squared values go up to 0.99, and that's where they are maintained through the suspension and working banks. So these tumoroid models, even as we're getting to later passage stages, still represent the integrity of the genome of the parent tissue. Importantly, they are not like each other. So this is both of our tumoroid lines, the 3309 line on the y-axis, the 1104 on the x-axis. This is again looking at the variant allele frequency across all the variants in these tumors. And at this point, these tumors are entirely genetically different. Our squared value is 0.11. As you can see, a lot of the variants are pushed against the axis. They are essentially exclusively either to the 3309 or the 1104 model. So two distinctly, completely different model systems just within these two side-by-side comparisons that we're talking about today. So in addition to all of that variant data, which is great, right? You can look for driver mutations, mutations for proteins that are going to be targeted. We also have started to look at RNA and protein expression as well. So what genes are expressed and are those various proteins expressed as well? So a lot of data on this slide, but if we're going to start on the left panel over here, again, our two donors, 3309 and 1104. Within each of those columns, we're going to look at the RNA or the protein, RNA on the left, protein on the right. So what we did for 3309 is basically said, of the RNA that is predicted to be expressed, so using poly-A-RNA-seq transcripts per million of over 1 predicted to be expressed, we then correlated that with mass spec generated protein data. So there were 5,000 genes that we identified where the RNA was expressed and there was protein expressed, and that's what's shown in these two columns on the left. We then took these RNA and protein genes and compared them to the 1104 line. And what I really like about this figure is that it shows that there are a lot of RNAs that were expressed in 3309 that are not expressed in the 1104 model, and it's even more drastic as you move to proteins. So not only, again, do you have distinct variants, you have distinct RNA and even distinct protein expression between these two models. And then we went ahead and did some confirmation studies. So we looked at two targets that are commonly actually targeted currently, and they're currently being explored for different therapies, which are HER2 and ROR1. And so in this case, we saw the presence of HER2 mRNA and protein in both models. And when we looked at this by flow cytometry data in these solid histograms, we can see that HER2 is expressed in both of these models, whereas ROR1 or ROR1 is only expressed RNA and protein in the 3309 model, and that was confirmed by flow cytometry. So now we have this really robust system that we can start looking at different targets on different biomarkers and how their expression changes with different proteins. What we really got interested in, though, in these models is, again, they're scalable. So we can grow up a lot of cells and we can start testing a lot of therapies, a lot of different concentrations, a lot of different therapies together to understand how these tumoroids are going to respond. And so looking at cancer therapies, we're looking at basically two large groups, one that is grayed out on the right. So we're going to focus first on the ones on the left, which are what we're calling tumor cell intrinsic. These are ones that are largely going to target the tumor cells themselves. Some of these aren't necessarily relevant for in vitro models, things like surgery and radiation, potentially not necessarily relevant, but the rest are. The idea of kind of a broad spectrum chemotherapy or some of these more targeted therapies directly against the tumor cells, be those antibodies or antibody drug conjugates or small molecule inhibitors. So what we started to do is a set of drug response studies that we're running on every tumoroid line that we're generating. These are 10 compounds that we're starting with. I expect this list to eventually grow, but what these initial 10 compounds are a mix of more standard chemotherapy compounds like paclitaxel, cisplatin, 5-fluorouracil, as well as a subset of small molecule inhibitors. And these are either broad multi-kinase inhibitors or a little more specific, like WEE1 inhibitors, PARP inhibitors, or those inhibitors against specific driver mutations, such as our KRAS G12C. For this webinar, we are also going to look at some ADC data, some antibody drug conjugate data. So since we knew that these two models expressed HER2, we did acquire 3 HER2 targeted ADCs. These do differ in the antibody used to target HER2. I will go through this a little bit more because we got some interesting data on this, as well as the payload used to deliver to the tumor cells. So what we've done here is basically use these compounds, cultured them with tumoroids for about a week, and then did a simple cell titer glow 3D assay, basically reading out luciferase activity as to percent of live cells still present in these cultures. So basically at the 0 concentration, so no drug added, we set that as 1, and basically as cells are dying, less live cells are present, and so we get nice dose response curves. As we increase dose of compound, we should see decreased live or increased death. So using our 2 cell lines, 3309 or 1104, again in blue and red, we saw that both of these died in response to 5-fluorouracil at relatively the same kinetics. Etoposide, there were some slightly different kinetics, but as we started to get to these small molecule inhibitors, we saw some differences. So in this case, for the WEE1 inhibitor, the 3309 line had a more enhanced response to that inhibitor than the 1104. What was very interesting is when we looked at the ADCs against targeted search HER2. So again, both of these lines express HER2 on their surface. We saw that by mass spec, we saw it by flow, but they had different responses. So in this case, basically all 3 lines or both lines didn't really respond to one of the HER2 targeting antibodies, whereas the 3309 did respond to the other. Now, what's interesting is this HER2 targeting antibody actually has a much higher affinity for HER2 than the other. And so it's really cool to see that that actually seems to have provided better binding and better transfer of this payload to this tumoroid line. But it also highlights the fact that simple expression of the target isn't enough, in that both of these express HER2, but really only one of them is showing a response to these ADCs. So there's other aspects of these tumors that we need to take into account when doing antibody drug conjugates. So the other side of cancer therapies are what we're calling tumor cell extrinsic. So these are ones where we're actually targeting part of the tumor microenvironment that is not the tumor. These also go into kind of 2 broad categories. This does say immunotherapies here, although we're going to make this a little more broad and say basically non-targeted therapies. So these are therapies that are trying to maybe induce a immune response within the tumor, using BCG, for example, to induce that in bladder cancer, versus those targeted therapies, but this time targeted to non-tumor cells. So the same type of idea, antibody drug conjugates, but maybe to a specific immune cell fraction or to the CAFs, the cancer-associated fibroblasts, or even down to some sort of adopted cell therapies or something like a bispecific antibody, trying to take immune cells and retarget them or reintroduce them to the tumor to elicit a response against the tumor. So what we're going to go through now is we've got our tumoroid system for cancer cells. Very, very good. It's very robust. What about the other components of the tumor microenvironment? So the first one that we focused on was cancer-associated fibroblasts, or CAFs. Great system to start with, worked on for decades at this point on how to culture these cells. And that's exactly what we did. So these are fibroblasts generated from both of our primary donors, our 3309 or 1104 models. In this case, that primary viably dissociated tissue was basically laid down on collagen-coated plates and allowed to expand. And what you get is these fibroblast lawns. They morphologically look like fibroblasts. When we look by flow cytometry for expression of markers that would be known to be expressed on fibroblasts, such as FAP or CD146, they express the markers that you would expect them to express. So very nice CAFs that could be grown out in addition. So now we've got the tumoroids, and we've got the CAFs from the same patient that we can start using to recreate the tumor microenvironment in an in vitro model. The next area that we were focused on, again, because it's been studied and there are some really great protocols out there on how to culture these, are the immune cell components. Now, some of these are a little bit easier than others. And so what we focused on first was the T cell component. T cells, highly characterized, very well described in vitro protocols, and also a lot of therapies targeting T cells within tumors to start looking at tumor eradication. So the one thing that we always take into account when we're starting to do these cultures is actually what the inherent microenvironment of the tumors look like before we start. And so we really like this graph because these are two indications that we see a lot that come into the lab, which is lung tumors and ovarian tumors. And what's really drastic about these tumors is that even though they're just epithelial-based tumors, they vary greatly within their immune cell composition. So for our lung tumors, tend to be highly immune infiltrated. That immune infiltration is very, very heavily skewed towards lymphocytes, B cells, and T cells, with a myeloid compartment, but a minor component of the overall immune component in the tumor. Ovarian is the complete flip. When we look at ovarian tumors, very, very tumor rich. A lot of times these are very immune cell poor. They're quote unquote cold tumors. And when we look at the immune composition of these tumors, while we do see T cells, B cells, and K cells, they're predominantly myeloid cells that are present in these tumors. And so again, as we start looking at therapies or the way to recreate these microenvironments in vitro, we also want to make sure that we're recreating them as most physiologically relevant to that primary tissue. So for T cells, what we've done here, so these are autologous CD3 positive T cells. So these are T cells from the parent tumor. We isolated out the T cells using a magnetic system and basically cultured them for 24 to 72 hours in the presence of either just a CD3 stimulation or a CD3 CD28 stimulation. And so what we're looking at here in the left panel is basically, can we induce these cells to proliferate and upregulate CD25, kind of a classic example of T cell activation. And so at the 0 hour when we start, or in the presence of no stimulation, we see no upregulation of CD25. We're using either CD3 or CD28. CD3, CD28 together, we can get robust activation or upregulation of CD25, as well as other markers like PD1. And these cells will robustly secrete cytokines, things like GM-CSF, interferon gamma. Interferon gamma is a great tumor eradicating or anti-tumor cytokine. So these T cells, we can get them out of these cryopreserved samples, we can culture them, we can activate them. They actually respond exactly like you would expect. So now we've got tumor cells, we've got CAFs, and we've got T cells all from the same donor. So the next component was actually, how do we start putting these all together and actually understanding what they're doing and getting a readout of response? And so to start this, we started looking at an IncuCyte S3 system. So we like the system because we can culture our tumoroids up to a week, which is generally about how long we like to go before we really need to, expand them or passage them. And every however long, I think we set this up to be about 12 hours, we're imaging each of these walls. So we can actually analyze the cells in real time. So what you're looking at here on the top is just one of our lines. I believe this is the 3309 line that is just growing in this culture system with nothing else added. And so we can see these nice round, very healthy looking tumoroids in this culture. On the bottom panel, what we added in initially was allogeneic T cells, or allogeneic NK cells, apologies. And I'll explain a little bit why we used allogeneic NK cells as we build out in our future considerations. But as you can see now, if we look at these, we see a lot more of these clusters, a lot more dark, almost necrotic centers. You could actually see the presence of these small NK cells as they're interacting with one of these tumoroids. So we're starting to work with this system a lot. We're trying to figure out ways to quantify this signal. We're also looking to remove the supernatants to look for cytokine secretion, other readouts outside of just bright field morphology as we're following these over time, but really starting to recapitulate this tumoroid microenvironment as much as we can in vitro. So kind of the idea of this part that Meredith set very well is we're now taking tumoroids, which is a tumor-only component, highly scalable. We're starting to add in the stromal elements, the CAFs, the fibroblasts from the same patient. Starting to take out these immune cells, particularly T cells that we can activate or reactivate in vitro, and trying to put these all together within one model system in vitro that we can now manipulate. We can add different immune cells, we can add different other cell types, we can add compounds or therapies and really see the responses. And so these are three of only obviously a ton of different cell types that we are looking at. And so future considerations that we are looking at in terms of how to really move this basically two broad groups. Autologous, those that came from the cancer patient themselves, and then allogeneic, those that came from a normal donor. So from the cancer patient, we have that primary tumor tissue. It is cryopreserved. We have tumoroids, we have CAVs, we have TILs, both CD3 TILs, as well as just the entire CD45 immune component. But from these cancer patients, we are also getting pre-surgery whole blood. So in addition, we have that peripheral immune component as well. This allows us to put a peripheral component back into these tumoroid or these ex vivo tumor microenvironment models as well. Obviously, there are some limitations just in terms of material availability with these systems. And so we're also looking at allogeneic as well, as it supports and gives more specifically immune cells to work with, but also there are allogeneic therapies out there that are going to be very, very interesting moving forward. And so we do have a robust leukapheresis clinic. From those, we are getting any variety of downstream derivatives, NK cells, gamma delta T cells. We like these two because those are not HLA-restricted immune cell populations, so they give us a lot of freedom when trying to do some co-cultures, as well as either bulk PBMCs, T cells, or specifically CD8 T cells. And so we are HLA sequencing all of our cancer patients, all of our normal donors, so that we can both match and mismatch HLA in terms of these co-cultures. One of the big things that's been difficult, honestly, for the last couple of years is the myeloid compartment. Myeloid cells traditionally are not great at being cultured. So what we started to evaluate here is bone marrow aspirates, again, through our normal donor clinic. Bone marrow can go through some differentiation to MDSCs in vitro. There's some nice protocols for that. So we're looking at a way of using this to actually reintroduce MDSCs or myeloid-deprived suppressor cells into the system. And then finally, umbilical cord. And so this became really interesting to us recently because we can get some of those other cell types that we can't necessarily get from blood to recapitulate the immune or the tumor microenvironment. Things like endothelial cells using HUVEX. Umbilical cord is very robust and very enriched for CD34 positive stem cells. Can we then use those to differentiate down multiple different pathways to also make additional cell types that might necessarily culture well from primary patient material or just be very, very difficult to isolate. So with that, thank you all for joining today. Love to take some questions. Both Meredith and I are available for those. Any additional questions that we can't get to today, or you have questions that come up later, always contact us at info@dls.com. I'll turn it back to Ryan for the Q&A section.
The very first question that I have for you here just wants to know, have you considered expanding the TILs from the parental tumor and cryopreserving those samples?
Yeah, I could take that one. So it's a great question. So we have done a lot of work on TILs where we have thawed and shown their function. That is the next iteration of this. Can we take those through a more broad, large-scale expansion and then really cryopreserve them back? That's the one thing we're always conscious of is scalability and reproducibility in these systems, because it is a finite starting system. So that actually is a big initiative for us for 2026. Meredith, I don't know if you have any other additional thoughts on our TIL expansion program.
Yeah, just thinking back to the slide you shared showing comparing lung and ovarian, right? And how, like, broadly speaking, lung is more T cell, B cell, ovarian is more myeloid. If the audience remembers, though, every dot there was an individual patient. And so there is, in addition to these kind of indication level trends, every unique patient it can go anywhere from 0 to 100 presence. And so something like taking these autologous tumor infiltrating immune cells, lymphocytes, and making those available is a way to speak to this real variability we see from one patient to the next.
Perfect. Thank you for that answer. The next question I have here for you wants to know then, for the tumor indications you are currently developing, the tumor indications you are currently developing tumoroids for are all epithelial derived.
Yes, that is correct. So our current focus is all epithelial derived. We have not started yet looking at things like glioblastoma, melanoma, things that are not necessarily epithelial derived. It's on the roadmap. I will say it's probably going to be some of the next iterations after we get those 6 in process developed, plus potentially a couple more. But at this time, all of our focus is on epithelial derived indications. Again, Meredith, any comments there?
Nothing else to add. Got it.
Perfect, thank you. The next question is then just wondering, do you have any plans to expand outside of those indications?
Yeah, so it is a big component of what we talk about. So of the, I guess, 10 that we have listed on that list, we regularly see broadly 50 plus indications if you factor in subtypes. And where we might say lung cancer, we mean lung adenocarcinoma or squamous, not neuroendocrine, right? So there is a big initiation to basically see as many of those indications as we can. How can we go ahead and get tumoroid models there? We have not broached outside of oncology at this point. I know it's something we've talked about, but we haven't broached it yet. Meredith, any comments there?
Yeah, so I'm a product manager, right? So I've always got my market intel hat on. We do have a form on our website where you can sign up to be notified for particular indications of interest. And also that data is used to help Sean prioritize, right? There are so many areas where we want to build this out to. And so if you are particularly interested in things like melanoma or non-small cell lung cancer or whatever, that is a place that you can show up and kind of vote almost to help get the lab priorities to match what your own project's priorities are.
Excellent, thank you. Now the next question is wondering, do you have any plans to expand outside of oncology into autoimmune or normal tissues?
Yeah, that's actually a great segue from the last question, which is I would love to. It's really a twofold at this point, which is the culture systems for those are going to be significantly different than the oncology ones we're developed currently. And then there's simply just the access to the tissue. I'll use autoimmune as an example here. We have some ability to acquire UC tissue, Crohn's tissue, some of the IBD tissues. Outside of that, we're finding it incredibly difficult to even acquire the primary tissue to initiate those for. And it's kind of the same way in the normal side as well. We have some cadaveric sites that we can work with, but cadaveric tissue has PMIs and things like that that we really need to take into account. So again, kind of like Meredith alluded to, it's on the roadmap. It's probably a later play at this point. Love to do it, just not quite there yet. Again, Meredith, any additional thoughts to add?
Just that in the meantime, blood is much easier to access from our autoimmune patient pool. And so, all of those immune cells present in the peripheral blood, that's, research that we can support today. And then, like Sean said, the tissue-based 3D cultures would be more of a long-term future play.
Thank you. The next question here is simply wondering, how long can tumoroids be passaged?
Yeah, so we've done quite a bit of work there. So what I've been calling the mid-passage banks there, those are generally somewhere in the 20 to 30 passages. Again, every time we generate a bank, we are making sure that it has maintained the heterogeneity of the parent tissue. So, and that actually goes to both genomic and transcriptomic. We haven't quite done that on the proteomic side yet. Those cultures have been pushed out 60 plus passages, half a year. We still haven't seen any drift there, but drift is always number one on our priority list, and we are checking it as much as possible to make sure it's maintained.
And then those low passage banks, right, that you talked about how, kind of the three phases to build one, we're freezing back some at every stage so that when we eventually do see drift, and like you said, it hasn't happened yet, but eventually we're expecting that to show up, that we can stop, return back to the low passage material and essentially restart the clock.
All right, perfect, thank you. Now, the next question I have wants to know, have you characterized extracellular matrix content in the tumor plus CAF plus immune cell 3D cultures?
Yeah, the ECM question is great question. So, full disclosure, when we're at that stage, a lot of times there is a little bit of ECM that we add just to encourage some aggregation of the sample. Conveniently, and one of the big reasons we got into the mass spec side of this is the lab next to us, we share a part with our proteomics team, which is both lovely and dangerous because we just chat all day. And they have been in our ear quite a bit about wanting to get material to ask that specific question. What is the ECM? Is there additional ECM being built? How is that compared to what we started with? So it's active part of what we're looking into. We haven't gotten there quite yet, but we've got all the teams together to make that happen.
Sean, any particular conferences to keep an eye on us being at next year, where we maybe would see that data?
Well, so
I see that he's making up an answer right now. This is not a Steve
yeah,
question.
we did not plan this one. We do have, so at AACR, we have submitted a proteomics tumoroid talk or poster. So there will be a lot of protein-based data there. And so that would probably be the nearest or the most recent that or upcoming that we would have some of that data available.
All right, perfect. Thank
you.
you.
Conference plans live.
Yeah. For the next question, someone's wondering, how can you identify the different cell populations like tumor cells, CAFs, and immune cells in a cell viability assay?
Yeah, so welcome to probably the conversation we have the most when we talk about these systems. The beauty of the cell titer glow system, right, where we're doing those drug response studies, it's quick, it's easy, get a lot of time points, a lot of conditions. It is 100% going to just lyse everything in there, right? So when we did those studies, right, that was only tumor cells, very focused on just that readout. As we're getting to these co-cultures, we are looking at several different systems. One is basically the addition of fluorescent caspase molecules to the InQ site to see if we can read out by morphology some specific caspase signals to look for cell viability. We're also looking at using flow as a readout. There we would have to disaggregate these, right? And so basically what you're going to end up doing is a lot of times is either looking for intracellular caspase activation, as well as the relative percentages of the components compared to the primary or an untreated sample. So it's something we talk a lot about in our tumoroid meetings. We played a lot with that in 2026, honestly, and hopefully, again, AACR time, mid next year, we'll have some data looking at specifically viability with each of those components.